Today we are going to see digestion and absorption of carbohydrate. Merely ingesting foods do not nourish you. Only after the nutrients content in the foods have released and it can be absorbed by bloodstream and distributed to all parts of the body. The major organs involved in the process of digestion and absorption are gastrointestinal tract, pancreas, liver and gallbladder. We can take our digestion and absorption system for granted because most of the process involved are autonomic, that is, they control themselves. We cannot decide when the pancreas secretes digestive enzymes into small intestine, what liver does with absorbed glucose, or how quickly food is propelled through the intestinal tract. Hormone-like compounds, nerves control these functions. Only two voluntary responses are decided: how well to chew foods and when to eliminate fecal remains. Thus, while you see about your daily routine activities like playing tennis, working in a laboratory, attending classes or eating lunch, many digestive reactions and activities occur automatically in your gastrointestinal tract. Let us understand these activities in this module. Learning objectives are: number 1, the physiology of digestion, important secretion of digestive tract, enzymes involved in digestion and types of absorption. Digestion and absorption are here. The ultimate goal of digestion and absorption of sugar and starch is to dismantle them into small molecules in the form of glucose that the body can absorb and utilize in the form of glucose. The large starch molecules require extensive breakdown. The disaccharides need only to be hydrolyzed once. The initial conversion of starch in the mouth and final conversion and absorption occur in small intestine. Then the conversion to a common energy currency takes place in the liver. When a person eats food containing starch, the enzyme hydrolyzes the long chain into shorter chain, the shorter chains to disaccharides, and finally the disaccharides to monosaccharides. This is the process known as digestion of carbohydrate. Starts in the mouth and ends in large intestine and rectum. Now we will see the physiology of digestion. The gastrointestinal tract is a long tube stretching from mouth to anus. This tube is otherwise known as alimentary canal. This is a complex system, performs a variety of physiological functions like movements, secretion, digestion, absorption, elimination, and nutrient production and absorption. Most of these functions are under autonomic control, that is, they are involuntary. Almost all functions involved in digestion and absorption are controlled by hormones, hormone-like compounds, and nerves. Now we will see the important secretion of digestive tract. Some of the secretion are saliva, which is secreted in mouth and used for the digestion of starch and also for lubrication. Mucus is another secretion which is secreted by mouth, stomach, small intestine and large intestine, used to protect cells and also used as a lubricant. Enzyme is a very important secretion secreted by mouth, stomach, small intestine and pancreas to promote digestion of food into small molecule for easy absorption from gastrointestinal tract. Gastric juice is secreted by stomach, which create acidic medium in stomach. The next one is pancreatic juice secreted by pancreas, which is essential to neutralize the acidic content of stomach. Now we will see the Enzymes involved in digestion of carbohydrate. The most important enzyme secreted in salivary gland is salivary amylase, which is essential to convert starch and glycogen to maltose and dextrin. Pancreas is another site which is capable of producing the enzyme called as pancreatic amylase, which is essential for digestion of starch, glycogen, dextrin into maltose. The intestine is capable of producing maltase, sucrase, lactase. These are able to digest maltose, sucrose, lactose by releasing glucose, glucose with a combination of fructose, fructose with the combination of galactose. The process of digestion. Let us begin by reviewing the main parts of the body involved in digestion. First, in mouth, salivary glands produce saliva. Saliva contains enzymes that break down carbohydrates to simple sugar, and mucus lubricates the food. Apart from this action, chewing divides solid particles into small manageable particles, which increases the surface area of the foods and making more efficient digestive action by the enzyme. Digestion of cooked starch by salivary amylase begins in the mouth, but food remains for too short period in mouth and hence very little digestion takes place in the mouth. The digestion of starch by salivary amylase continues in the stomach for 10 to 15 minutes till the food gets mixed with gastric juice and the action of amylase is stopped in acidic environment of the stomach. Vigorous chewing of high fiber foods slows eating and stimulating the flow of saliva. Saliva enzyme amylase starts to work hydrolysing starch to short polysaccharides unto maltose. The mouth and stomach are connected by esophagus. As it enters a valve-like flap of tissue, epiglottis, it is present that helps to prevent food from being loaded in trachea, that is windpipe. When food is swallowed, it landed on epiglottis, which then covers larynx, that is a opening of trachea. The breathing automatically stops. These involuntary response ensure that the swallowed food travels only down the esophagus, helped by muscle contraction of esophagus and by cavity, and the food reaches the stomach. The stomach is basically holding tank with a capacity of nearly 1 liter. The stomach continues the process of digestion by enzyme and acidic gastric juice. As the result, the semi-solid mass of food with these secretions are obtained and are known as chyme. This chyme readily leaves the stomach within 2 to 4 hours after food is eaten. If the chyme is more solid, the longer it takes to leave the stomach. The chyme high in fiber content generally takes longer time to leave the stomach than chyme high in carbohydrates. Fiber in the stomach also delay gastric emptying, thereby providing a feeling of fullness and satiety. The liver, pancreas and gallbladder work with GIT but are not physical part of it. The liver provides bile, which aids in fat digestion and absorption and is stored in the gallbladder until needed. The common bile duct leads from the gallbladder and continues with pancreatic duct, which allows digestive enzyme and other products from the pancreas to be mixed with bile before entering duodenum for digestion. To a small extent of stomach acid continue breaking starch down, but its juice does not contain any enzyme to digest carbohydrate. Stomach empties into small intestine which is coiled below it. In further breakdown of carbohydrate takes place. The small intestine is divided into three sections. The first part is duodenum, is about 0.3 meter long. The middle segment and the jejunum is about 1.3 meter long, and the last section ileum is about 1.6 meter long. The small intestine is the main site of digestion of carbohydrate and also considered small because of its narrow diameter, that is 2.5 centimeter. The pancreatic duct opens into duodenum. The alkaline neutralizes the pancreatic juice. This neutralization of this acidic content of stomach and change the pH and activates the activity of enzyme. The pancreatic juice has mixture of carbohydrate digesting enzymes like pancreatic amylase and enters the intestine via pancreatic duct and continues breaking down the polysaccharides of glycogen and starch to disaccharides like maltose. The final step takes place on the outer membrane of intestinal cells. These specific enzymes dismantle specific disaccharides. Thereby, most digestion is completed in the duodenum, upper jejunum with the help of enzyme from intestine and the pancreas. The muscular contraction in small intestine constantly mix the food with digestive fluid and enhancing digestion. A small meal remains in the small intestine is for about 3 to 10 hours. At this point, the following simple sugars are produced. For example, maltose is a disaccharide with the involvement of the enzyme maltase produce 2 molecules of monosaccharides like glucose. Sucrose is another disaccharide produce 2 monosaccharides in the form of glucose and fructose with the involvement of the enzyme called as sucrase. Lactose is a milk sugar capable of producing 2 monosaccharides in the form of glucose and galactose, and the enzyme involved is lactase. Dextrose is another polysaccharide produce dextrin with the involvement of the enzyme known as dextrinase. The small intestine empties into large intestine or colon. This organ is about 1.1 meter long and is separated into 5 sections: the cecum, ascending colon, trans colon, descending colon, and sigmoid colon. Within 1 to 4 hours after a meal, only small fraction of starch and indigestible fibers remain in large intestine. The small fraction of starch that escapes digestion and absorption in small intestine is known as resistant starch. Starch may resist digestion for several reasons, reflecting both the individual efficiency in digesting starch and physical properties of food. The resistant starch remain in large intestine, promotes bowel movement as fiber do, but unlike fiber, the resistant starch do not lower blood cholesterol level. Fiber and resistant starch in large intestine attract water and soften the fecal matter for easy passage without any staining. Bacteria in human digestive tract ferment both fibers and resistant starches. This process generates water, gas, and short-chain fatty acids. The short-chain fatty acids are absorbed in colon and yield energy when metabolized. Metabolism of short-chain fatty acids occur in both intestinal mucosa and liver. Food fibers and resistant starch do contribute some energy, about 2 kilocalories per gram. depending on the extent to which they are broken down and are absorbed. In general, foods that reach the large intestine is mostly indigestible. This residue remains in large intestine for about 24 to 72 hours before elimination from the body as feces. The terminus of large intestine is attached to rectum, which is connected to the anus. These final section of gastrointestinal tract work with large intestine to prepare the feces for elimination. When either the small intestine or the pancreas is diseased, inadequate quantities of important digestive enzyme may be produced. This scarcity can result in incomplete digestion and very limited absorption. In such cases, nutrients in undigested food travel into large intestine rather than being absorbed into the bloodstream. In large intestine, the undigested food is metabolized into acid and gas by bacteria. The resultant feces appear foamy and greasy due to trapped gas and presence of undigested fat. Intestinal malabsorption often causes a distended abdomen due to intestinal gas. Whenever digestion and absorption are hampered, the body pays the price. Insufficient enzyme production or insufficient time to complete enzyme action is often the cause of these problems. The resulting monosaccharides, majority in the form of glucose with fructose and galactose, are absorbed in small intestine. Some amount of pentose present in food are liberated from the digestion of nucleic acid or absorbed for utilization. Now we will see the absorption of monosaccharides. Absorption is the process in which nutrients molecules are absorbed by gastrointestinal tract and enter the bloodstream. Most of the nutrient absorption occurs in small intestine. Stomach and large intestine participate to minor extent. The extent and efficiency of absorption in small intestine are linked to its incredible surface area. The wall of small intestine folded, and within the folds finger-like projection known as villi are found. The finger trap nutrients between each other to enhance absorption. Each villus finger is made up of numerous absorptive cells. Each of the cell has brush border or microvilli covered by hair-like projection called glycocalyx. Intestinal enzymes are often found on glycocalyx. All these folds, villi in the small intestine, increase in surface area 600 times beyond that of small tube. Now we will see the types of absorption. The small intestine uses 4 basic types of absorptive process. One is passive, facilitated, active, and endocytosis. The passive absorption occurs when nutrients enter absorptive cells without a carrier or energy expenditure. For this to happen, the walls of the intestine must be permeable to the nutrients, and nutrient must be present in a higher concentration in the intestinal lumen than in absorptive cell. The difference in concentration derives passive absorption. Water, fat, and some minerals are passively absorbed. In this simple diffusion, the sugar molecule diffuse freely across the intestinal membrane down the concentration gradient. From the intestinal lumen, where the concentration of a particular sugar is higher than the circulating blood, where the concentration is low. Hence, passive absorption requires permeability of the substance through the walls of small intestine and a concentration gradient higher in intestinal content than in absorptive tube. Facilitative absorption. Facilitative absorption uses a carrier molecule to shuttle the nutrients from lumen of small intestine into the absorptive cell, but no energy is expended. Again, the concentration difference derives the reaction. The facilitative absorption takes place for the simple sugar fructose. The nature of the carrier molecule is in question. In this facilitated absorption, the sugar diffuses down concentration gradient across the membrane more rapidly with the help of a protein carrier like passive diffusion. This diffusion does not require metabolic energy. The third one is active absorption or diffusion. It uses a carrier and the process requires energy. A single sugar, glucose or galactose and other nutrients are actively absorbed. ATP is the energy source. Using this energy, absorptive cells can take up a substance in lower concentration in the intestinal content and move it into the cells where the concentration is higher. Since the bloodstream constantly bathes the absorptive cells, their native concentration of glucose is higher than that of the intestinal content. Therefore, the ability to absorb against a concentration gradient is critical. A second type of active absorption involves the process of endocytin, phagocytosis, literally cell eating, and pinocytosis, cell drinking. In endocytosis, an absorptive cell engulfs compounds or liquids identity its cell membrane. When particles or fluid move into the indentation, the cell surrounds them. An infant absorption of antibodies from mothers will occur this way. In general, carbohydrate absorption is discussed with reference to carbohydrate-rich foods like banana and bread. In this preparation, sugar occurs naturally in the fluids or added as table sugar and are formed by the product of starch digestion in the mouth and intestine. Fructose is taken up by the absorptive cells of villi along with the sodium during active absorption. The energy used in the process is actually needed to pump the sodium ions back out of the absorptive cells. It is important to remember that enzymes essential to carbohydrate digestion originate both from pancreas and from cells of intestinal wall. The intestinal disease can interfere with the production of enzymes formed in intestinal wall. Such condition may interfere with the efficient digestion of sugar like maltose, lactose and sucrose. If these carbohydrates are not fully digested, they are not absorbed. Instead, they reach the large intestine. The resistant bacteria uses the sugar to produce acid and gases, causing abdominal discomfort. People recovering from intestinal disorder such as diarrhea or bacterial infection may need to avoid lactose for few weeks because of the temporary lactose intolerance. In 2 weeks, the small intestine begins producing enough lactase to permit lactose digestion. Large intestine absorb primarily sodium and potassium along with some water, leaving only about 200 ml of water unabsorbed. This occurs mostly in first half of large intestine. Short chain fatty acid made from both metabolism of some plant fibers and undigested starch are observed in large intestine along with some vitamins synthesized by bacteria such as vitamin K and biotin. By the time the content of large intestine passed through the first 2/3 of its length, semi-solid mass is formed. This remains in large intestine until peristaltic wave and mass movement pushed it into rectum for elimination. Once glucose, galactose and fructose enters the villi, they are transported via portal vein to the liver for glycogen production, fat production, energy use or direct release into the blood. Now we will see the conclusion. The breakdown of foods into smaller units and finally into absorptive nutrients involve both physical and chemical processes. The physical process comes first as food is broken down into smaller pieces. Chewing starts the breakup and muscular contraction of gastrointestinal tract continues it. As GIT breaks up food, it mixes with the food with various secretion and moves the mixture along with the tract. Enzymes along with other chemicals helps to complete the breakdown process and promote the rate of absorption for utilization of nutrients to optimize the health and well-being of an individual.